to today's episode of Myeloma Crowd Radio. I'm your host, Jenny Alstrom. We'd like to thank today's episode sponsor, Takeda Oncology, for their support of Myeloma Crowd Radio and all that they do for multiple myeloma patients. We have two announcements today before we get the show started. The first is that we're introducing Myeloma Crowd TV, a monthly episode that will air on the Myeloma Crowd YouTube channel and on the Myeloma Crowd website. This is a program for patients, by patients, on topics that matter to you. So you can check out our first episode. That's a great review of the exciting announcements from last year, and you'll find that on the Myeloma Crowd website. Our second announcement is a program we just barely opened up called Muscles for Myeloma. Now, at ASH, we heard a lot that myeloma specialists are segmenting patients into fit, unfit, and frail categories and are tailoring treatment based on those categories rather than by age. So this is a fitness challenge to get us all up and moving while helping a good cause, myeloma research. Now, patients' ability to handle a more difficult treatment or extended treatments also typically gives them longer overall survival. So even though you may not feel up to it, any exercise plan is better than no exercise plan for myeloma patients. So here's how it works. You can click on the link on the Myeloma Crowd site in the top right corner and create your own Muscles for Myeloma page where you can tell everyone how you plan to get fit between March and April of the year. Now, add a photo and even a video if you'd like. You can invite your friends, family, or coworkers to join you and create a team page. And then to add some accountability, you can ask your friends and family to sponsor you for each minute you exercise. It could be 1 cent per minute, 10 cents a minute, $1 a minute, anything. Let your supporters decide. It's like the elementary school walk-a-thon, but for myeloma, and all proceeds will be donated to the Myeloma Crowd Research Initiative, two important immunotherapy projects that are dramatically changing myeloma care. So now I'm so excited about this because exercise is really what got me out of my myeloma funk, I would call it, after treatment. It's truly what kick-started me into being a patient advocate. So even we're just coming out of transplant or other treatments, there is something we can do to work on better fitness. And of course, you should consult with your doctor before starting any program. Now on to today's show. We are delighted to have Dr. James Berenson with us to discuss very important work he's doing on a way to track myeloma's absence and growth. Welcome, Dr. Berenson.
Thank you. It's my pleasure to be with you today.
Well, we're thrilled to have you. So let me introduce you before we get started. Dr. James R. Berenson is Medical and Scientific Director and President and Chief Executive Officer of the Institute for Myeloma and Bone Cancer Research in Hollywood, California. He's also President and Chief Executive Officer of Oncotherapeutics. Dr. Berenson is a member of many committees and societies, including the ABMTR Multiple Myeloma Working Committee, the Scientific Board of the International Myeloma Foundation, and the MMRF. He's on the Medical Advisory Board of the Lymphoma Research Foundation, the Myeloma Working Committee of the Autologous Blood and Marrow Transplant Registry, and many others. Dr. Berenson has been on the Who's Who list, including the Sterling Who's Who, the 2000 Outstanding Scientists of the 20th Century, the Madison and Emerald Who's Who lists, and Distinguished Professionals Who's Who lists. He is also a member of the IMBCR Working Group Myeloma Committee, as well as a member of the Leukemia and Lymphoma Society Foundation's Board of Directors. He's editor for publications including the Annals of Hematology, Annals of Oncology, British Journal of Hematology, Clinical Advances in Hematology and Oncology, Targeted Oncology, and many, many others. So, Dr. Berenson, thank you so much for joining us on the show today.
Thank you.
Now, when you were in Utah, we had an opportunity to get together, and you had a chance to ski a little, which hopefully you had a great time here.
That's where I did my training over in Salt Lake. Beautiful place.
Well, it's perfect. And we're very grateful that you're doing the work that you are, but we were talking about something that I thought would be very, very interesting for patients to understand. And I guess we'll start by understanding first how myeloma is currently measured and what tests are typically used on an ongoing basis to track disease, because this is what you're working on.
Yes. So in myeloma, we don't actually have an ability to measure the tumor because it's throughout the bone marrow. So unlike breast, lung, colon, where you can get your hands, if you will, around the size of the tumor, since myeloma is throughout the bone marrow, which is the inner part of your bones, we indirectly follow this by measuring blood levels and urine levels of a protein product, which is the antibody. Since this is a cancer antibody-producing cells, and normally one plasma cell, the antibody-producing cell, the malignant culprit in myeloma, but normally a plasma cell makes one antibody, and in those without myeloma, we make billions of different types of antibody. But what happens in myeloma is too many of these plasma cells gather in the bone marrow, and they spew out and secrete into the blood and sometimes into the urine the monoclonal protein. This is an antibody, a protein, and it can be found in the blood and it can be found in the urine. In the urine, when it's found there, it's usually part of the antibody, and we measure that as a whole antibody, which consists of two long chains called heavy chains, which can be C alpha most commonly or C gamma. Those two are most common. That's for IgA or IgG. Uncommonly, you can see C delta or C mu. That's for IgD and IgM, and very rarely C epsilon, which is for IgE. So that's a heavy chain component. Then you have two shorter chains, and those are called light chains because they're lighter in weight. They're about half the length, and those can be either lambda or kappa in myeloma. Just like with normal people, there's more kappa than lambda myeloma. So it's been said these are abnormal antibodies. That's not actually true. They're normal antibodies, but they're being put out by an abnormal malignant plasma cells. So what happens in myeloma, unlike normally when you have billions and billions of different antibody-producing cells making trillions of antibodies, in myeloma you have a clone that takes over and you only make one antibody, and that's the M protein, which in full force is an entire heavy and light chain, so like an IgG kappa or maybe an IgA lambda. But in some folks, they don't make the heavy chain, they only make the light chain component, so they're kappa or lambda light chain myeloma. And then that slips into the urine. So in a normal kidney, the whole antibody is too big to get through the membrane into your urine, but the smaller one can fit through, and therefore you find it in the urine. We call that Bench-Jones protein, and we measure that as 24-hour amounts of urine protein and then figure out what part of that urine protein is the abnormal component. Now, in the last few years, the group at Binding Site has developed a way to measure that same light chain in the blood, the so-called serum-free light chain assay, and that is done as well to monitor disease. So changes and decreases in those levels suggest improvement in your myeloma, and increases suggest things are going poorly with the myeloma. The other thing that's done to monitor myeloma is to do bone marrows and radiologic tests and those sorts of things. Those are kind of difficult to follow in a typical myeloma patient. So monitoring myeloma with bone marrow tests, because myeloma is so, if you will, variably involved in the marrow. You can see spots where it's 10% myeloma, and other spots it's 50, other spots are 1. You generally can't really guide your treatment with percentage or proportion of myeloma cells in the bone marrow. So we don't typically do that. We do that in patients to diagnose them when they're required to have bone marrows for clinical trials, or if their blood counts are very low and we're not sure is it the myeloma or is it the treatment that we overdid and wiped out their bone marrow function, then we'll do marrows. The marrows are not that great. Now, radiologically, there's been a great increase in the number of MRIs and PET CT scans, and I've seen a bunch of patients getting those today. And we generally still perform bone survey, that's plain X-ray of the bones, and we do not routinely do that as follow-up because many times the radiologist will imagine new lesions that are not real. You have to be very careful. And MRIs we'll do in patients who have particular areas of symptoms, but we don't routinely do that, and the same for PET CT scans, except for under specific circumstances. So the tests that we generally use to follow myeloma traditionally are blood and urine levels of the protein over time. Now, it turns out the amounts of those at the time you're diagnosed don't really prognosticate. They don't really predict how well you're going to do. So there have been attempts with other staging systems with other proteins to tell how long you're going to live with your myeloma. I would tell you the good news now is people are living so long in our clinic, those particular staging systems are not useful anymore. So we've relied on first the Dury Salmon and in the last 10 years on what we call the international staging, which is simply based on the blood levels of your healthy serum albumin and, if you will, your unhealthy beta-2 microglobulin, which is on the myeloma cell, and when shed in the blood, then you can measure it. So if you have more myeloma, you have higher levels of this beta-2 microglobulin level in the blood. So those tests have been used, the two of them, to say you're stage 1, 2, or 3, but I would tell you we've just analyzed about 300 patients in our own clinic. There's no use to that anymore. That has not been a useful staging system. So prognosticating myeloma with what we've had to date in the last few years, thanks to all the new drugs, is not very useful.
Well, it's good to know. I mean, it's hopeful if you were a stage later that it might not be indicative of how you'll end up.
Yeah, and one of the reasons for that, Jenny, is that we have now, thankfully, so many good drugs to treat kidney myeloma. You come in with myeloma that's mucked up and your kidney function is poor, and we know that this beta-2 microglobulin protein is affected by kidney function. So even if you have diabetes or high blood pressure causing kidney problems, you're going to be a stage 3, but it's not from myeloma. But these days we have such good treatments for myeloma bone disease that we can, in most folks, reverse it. So those people who used to have stage 3 and do poorly, now with stage 3 can readily be reversed because of the effectiveness of the new drugs we have in 2016.
And in your opinion, if the staging system, the Salmon Dury and the ISS staging systems are kind of outdated, I guess, with the newer drugs, what do you think would be a good replacement?
Well, I guess that'll segue me into talking about our new marker, which has not certainly been proven 100% to be there, but we're on our way. So in the last several years, we've discovered a new protein that is in the blood of myeloma, other leukemia patients, lymphoma, and now we're even on to immune deficiency patients. So this is a protein which is being targeted by many companies now to treat myeloma because it's present on late B lymphocytes, the type of cell that's the malignant culprit in myeloma, again, the plasma cell. And what we've shown is that it's shed in the blood, and the more myeloma, the higher the level, and we now know with certainty that the higher the level, the worse your outcome is going to be, not only in terms of how long it's going to take you to progress from your present treatment, but actually how long you're going to live. It really does predict outcome, that specific marker.
Okay, and this is I think patients may have heard about this marker before just because other companies are doing work targeting this. So do you want to go into what that protein is and then maybe what it does?
Yeah, so that protein is called B-cell maturation antigen, and it was found and identified because in certain kinds of rare lymphomas, they had cloned out this protein 10, 15 years ago, and then it was shown that that protein was present on late B cells and also on malignant plasma cells as well as normal plasma cells, so that when this protein was bound by certain factors, one's called BAF, another one's called APRIL, this would drive the growth of the myeloma and normal development of late B cells and plasma cells to make antibody. So this is a normal component of late B cells, but happens to be present in large amounts on malignant plasma cells as well. So in the last couple of years, several companies that we and others are working with are actually targeting this same protein for therapy, thinking that if they can block it, that receptor, they'll stop the growth of myeloma, or in some cases, they're using toxins that are hitched onto the back of an antibody, which is delivered in high concentration to the myeloma, because the myeloma has that tagged marker BCMA on it. You bring the antibody, and then you dump truck the toxin, you kill the myeloma. So there is a big, huge interest in BCMA as a target for treating myeloma in the last 12 to 24 months now.
And a question, you mentioned that it's on late-stage B-cell lymphocytes. Is it on early type of, and on malignant cells? So would that mean it's on all myeloma cells, including early stage and late stage? I know sometimes the doctors will talk about the stem cell, basically, or where the cell that's creating problems after even the late stage stuff is killed.
Yeah, I don't think we can tell you with certainty yet that it's present on a stem cell population. I think it probably is, but I'm not going to make that leap of faith yet. But I think it probably is present across the spectrum of early, if you will, the ever-ready battery cell that you can call the stem cell. I call it the ever-ready battery cell that just keeps giving you the myeloma that we need to eliminate. I think it's there as well.
Mm-hmm. And do you want to explain, you did this very well when we met in Utah, do you want to explain how BCMA tracks with the M protein, or what you found when you were running these tests?
Yeah, so that's a really good question, Jenny. So what is the advantage of this potentially new test we've developed? Well, the beauty of this is that BCMA turns over in your blood every day, whereas some of you may have heard of the new monoclonal antibodies interfering with your ability to detect complete remission. That's because antibodies, whether the treatment antibodies like Darzalex, daratumumab, elotuzumab, Implicity, these antibodies hang around a long time. Their half-life in the blood is 3 to 4 weeks. So I told Jenny it's kind of like if you're looking at BCMA, which is sitting outside my window, I'm looking at our sun, and so I get a very quick read, whereas with conventional markers, because the turnover of IgG is so slow, it's like you're looking at the light of the sun that's about 4 galaxies away. So we believe with our new marker, since the turnover is so fast, we can get a read, and we already have data on this, within a few days to tell you whether a drug is or is not working. So you don't have to remain on therapy for 2 months getting side effects and getting treatment that isn't really working. You should be moving on, but we can't tell you that because we haven't been able to get a marker that can turn over that fast, and we now have it with BCMA. The other advantage that we have with this, there are a subset of patients, and there are more and more evolving over time that become either non-secretory or, for example, one of my favorite patients from the Bay Area who started non-secretory, but a lot of people become non-secretory over time. And what that means is they do not make the conventional marker, the IgG or the light chain marker. They don't make anything. We've had no way to follow them. And with BCMA, we have a really great test. We've shown that this test correlates with bone marrows that these people have to have every few months, or PET CT scan activity. We now have a blood test to follow them, which is just great because they haven't been able to be followed up until now.
Well, yeah, and no one wants to do, bone marrow biopsies every few weeks to check on your disease status. That's not good. Okay, so, and also when you were showing some of the charts that you had, it looked like the BCMA tracked almost identically to the M protein. So that's what gave you the idea that you could use this as a supplement or even as a replacement.
Yes, that's correct. It does track faithfully with conventional markers, but one of the things we've noted is that with the free light assay, there's a lot of variability in the results, and those results are very hard to interpret with anybody with kidney disease. And this test beautifully, unlike a lot of other tests, runs totally independently of renal function, of kidney function. So you can faithfully look at this whether your creatinine's 5 or whether it's 0.5. And that's a huge advantage compared to some of the other tests we're using to monitor myeloma right now. And, for example, 24-hour urine protein is only as reliable as the patient is to gather it up. A lot of patients don't accurately collect it. By the way, we've shown this is also in the urine, although we're not using that right currently to monitor patients. But 24-hour urine proteins are really a lot of times inaccurate and change also with kidney function, the results of the M protein.
Okay, and then when we were meeting, you said something about BCMA at too high levels and then BCMA at too low levels, and that might give you an indication of how you might do. So do you want to talk a little bit more about how you can use this as a prognostic tool?
Yeah, we're just publishing the data now. It's a really cool story. So what we noticed, what you probably all know is, as a myeloma patient, you generally don't make normal antibody. And for many years, it's been known or thought that there is a protein or soluble substance in the blood that suppresses your ability to make normal antibody. So you make all this one antibody, you don't make anything else. And we've now figured out what the culprit is. It's BCMA. So what happens is when BCMA is shed into the blood, it soaks up those factors I mentioned earlier, BAFF and APRIL, which are the fertilizer to drive your normal B cells to make plasma cells to make antibody. So basically, we're sequestering the fertilizer that drives your immune system when you have BCMA at a high level. So therefore, you don't make antibody. And then what we noticed is in people that were in complete remission, everybody has BCMA because everybody makes plasma cells. So the normal levels run about 35 to 40 nanograms per milliliter. And what we noted, somewhat surprisingly, that complete remitters, half of them had very, very low BCMAs, and half of them had levels that were 35 range. And then when we look further, the ones with extremely low BCMA, they don't make antibody because they don't have plasma cells. And the ones that actually have normal BCMA levels make normal antibody. So we've gone on now to show that this is a marker. In fact, I have a call as soon as this is on. We're writing a large grant now. We've shown it's a beautiful marker for patients with immune deficiencies without even cancer. Kids with the bubble boy type, those with like the thing that led to the ibrutinib, they have this mutation, what's called the Bruton tyrosine kinase. Those people have no BCMA and they don't have any plasma cells. So we have a monitor now for cancer of the immune system and too little of an immune system, and it's the same darn marker. The other thing that's really cool about BCMA is it not only is a monitoring device, it's again a predictor of outcome, and I've never seen one marker that really can do both of those, and we can do that with BCMA. We can actually mark your myeloma, prognosticating your outcome, but we can also use it to monitor you.
Okay, so people with normal BCMA are doing best. To summarize your results.
Jenny, you asked a great. That's the leap of faith. We hope we're going to have enough data to say that. We don't quite have enough. We have a nice, strong trend that way, but we need more follow-up. But it appears that if your BCMA and you're in complete remission is really low, that is not such a good thing. You'd rather have your BCMA. I saw a physician patient from Fresno earlier, and I was cheering him along. He was on RVD in complete remission, and his BCMA was 23 in the normal range. I'm like, that's really good news, and you're probably making normal antibody. Whereas if your level is 3 or 4, you probably have an impaired immune system, and that may not be such good news. It also may mean that because you don't have a good immune system, you're getting a lot of infections, and we're seeing that with the story with immune deficiency patients. So if they have really low levels, those are people that seem to be at higher risk to get a lot of infections.
And then what happens to patients who have BCMA that's off the chart high? Or, how much over the normal level does it have to be?
Yeah, that's a great question. What happens to people who have sky-high BCMAs? Not a good thing. So if your BCMAs are in the top quartile, the top 25%, when you walk in, that's not a sign you're going to do very well. High levels are bad. We also know that the level that you have predicts how deep your response is going to be. So if you come in with a level of 50 instead of 5,000, you're much more likely to achieve a complete remission than the guy that walks in with 5,000. So unlike IG levels or M protein, which does not predict the depth of response, whether you have a 5 or a 0.5, this level does. The higher, the less likely you're going to have a good response.
Well, that would give you so much more information about your disease state, I would think. But rather than just, I have this monoclonal protein and it's X amount, which might tell you a little bit about your disease burden and maybe some about your bone damage. But have you seen any relationship with bone damage to BCMA?
No, we looked at that, Jenny, and we really didn't see. We showed that was really independent of bone disease. So I can't tell you this is a bone marker per se at this point. We don't have that data. It probably isn't, but that's actually good in a way because it predicts your outcome irrespective of bone disease, kidney disease, hemoglobin, conventional staging, age, sex, all that. It's independent of all that.
And so you made me curious. Is there any way I can go back at my labs or other patients can go back at their labs And is BCMA tracked anywhere in the standard labs that we've already had done, most likely?
No, this is not yet commercially available. We are developing that as a commercial assay. I would expect that to be true by the end of the year, although we are now wrapping it into multiple pharma and academic trials. So we're collaborating with the Mayo, UC San Diego, the Farber, the Italian group, and trying to spread the love about BCMA, and now we've embedded it into trials with all of the new drugs that you could think about. Each company now seems to be coming on board, and there are multiple companies, as Jenny mentioned, that are targeting BCMA to treat myeloma, so they're especially interested to seeing whether this will predict responses to their anti-BCMA therapeutic approaches.
Yeah, I guess that's my next question. So let's say you have too high of BCMA and you test for it, and then you knock out the BCMA with one of these BCMA-targeted treatments. Then you said if you go too low, that's not good either, because then you're not developing plasma cells. So how does that work? Is there any concern about knocking out all BCMA, or?
Well, I think you ask a really great question, and the good news, and it's really preliminary, but what we're seeing so far, the density of BCMA on myeloma cells appears to be much higher than on normal plasma cells. So we may be able to knock off the myeloma and not as much impact your normal cells. We don't know that for sure, Jenny. I certainly admit that to you at this point. I would also tell you, though, interestingly, that one of the approaches we want to take is, because BCMA is shed in the blood, there are two bad things that may happen if you're going to use an anti-BCMA approach. One is there may be so much of it in the blood you'll never get the antibody to the plasma cell. It's all going to be soaked up in the blood, and therefore you'll never be able to use it effectively to kill the myeloma. Two is, if we can keep it on the plasma cell, it will be a better target because there'll be so much density there. It'll be much better for the anti-BCMA to come in and bind and do its thing. And then I mentioned earlier that when this BCMA is rolling about in the blood, it's grabbing up all the fertilizer that drives your immune system, so we obviously don't want that to happen. So we want to prevent the shedding. Well, it turns out there's an enzyme called gamma secretase, which actually clips this guy and lets it go into the blood. So we're now in the throes of using blockers of gamma secretase to see if we can keep BCMA on the myeloma cell and prevent it from going in the blood, make it a better target, prevent the immune deficiency, and hopefully those anti-BCMA approaches will work better.
Okay, so what you're saying is that sometimes it's in the blood, and because you need to get it to the bone marrow as well, it gets used up basically when you treat the blood with high concentration levels in the blood that can never get to the bone marrow. Is that what you're saying?
Yeah,
with some of the BCMA-targeted treatments? Okay.
Yeah, that's possible. I can't tell you we have any data at all because we're just beginning those trials now with anti-BCMA antibodies. We're really early.
Yeah, I know there are some monoclonal antibodies to do that. There are also CAR T-cell targets targeting the BCMA. So do you have any insights in how it relates to that?
Well, I tell you, it could be the same problem if it's circulating. We'll just have to see what happens. That was my first work in oncology back in the early 70s, which showed that T cells kill tumors back in about 1973, a while back. But now we're actually doing it therapeutically.
Mm-hmm. Well, it's exciting and tragic that it takes so long, but exciting that it's coming.
Yeah, it's coming.
So when you talk about development of a test like this, how long does it take to develop something like this?
Well, we hope that this will be able to be obtained and from your doctors in a commercial assay within the next 9 to 12 months. It's certainly much faster than drug development. As you know, this is not a drug. It's a assay to measure and monitor the disease, so it's quicker. But it will be probably by the end of this year. I think we'll be able to have a test that you can use routinely to monitor your disease.
And like you said, it's just for blood. It's just a blood test.
Well, it's in the urine too, but we haven't developed that far enough along yet to tell you how to monitor that. It's definitely in the urine.
And for your patients in your clinic, are you using this test for everyone or just in a clinical trial environment, or how are you using it?
No, we're routinely using it, but I must tell you, obviously, this is not an on-label test, and so therefore you can only use it as a research-only test at this point. But it certainly is telling us a lot about what's going on with our own patients.
Mm-hmm. Well, I have friends who are non-secretors, and I think especially for them, I don't know what percentage of patients have that or become that over time. But
well, it's several percent present
mm-hmm.
with it, and probably up to 10% over time. And a lot of people become oligosecretory, and they stop making antibody over time. And people always say to me, well, my IgG is only 900. When I first came in, it was 3,000. But it's because over time you get the cells become less productive of the marker. That happens over time.
So when you hear IgG, because most people know I'm IgG kappa or IgA lambda or whatever, but those have never really had very good prognostic indications. Like you don't really know how you're going to do if you have one or the other of those. Generally, I think there might be some that are more kind of higher risk, I guess, than others. But does BCMA, or do you find it, I guess, in like IgG more kappa or in the lambdas?
No,
or
it doesn't.
IgA or IgG?
That's a good question. It doesn't correlate nicely with any of that, thank God, really. So it doesn't correlate with whether you're IgG or IgA or IgM or whatever at all. What we do know, however, as I mentioned earlier, that if you have an IgG myeloma and your BCMA is really high, your IgA and IgM are going to be on the floor. And that's, again, because the BCMA is binding up the factors that drive your normal immune system. You can't do that. But it doesn't correlate. It correlates with the level, as I said, but it doesn't correlate. Isn't higher if you're an IgA than an IgG. None of that correlates.
Mm-hmm. And let's talk for a minute about the status of your immune system, because now that all these immunotherapies are coming out, I've had doctors say, we need to not only know your genetic features to treat your myeloma, because there might be certain drug combinations that are better or certain treatments that are better for you with particular genetic features, but you also have to understand the condition of your immune system. So how does a patient go about understanding the condition of their immune system? It sounds like BCMA might have an impact there.
We think it might. We hope it does, on both ends of the spectrum. We don't know for sure. In terms of monitoring your immune system, we're pretty simplistic about it now. We measure lymphocyte counts. Sometimes you can measure what's called certain types of lymphocytes, certain types of T cells in your blood, or natural killer cells. And we're really at its early stages of figuring out how to monitor immune systems of patients in a clinically meaningful way. We can do tests, but in clinically meaningful ways, we don't necessarily know how to do it. It's clear that myeloma patients have a woefully inadequate immune system, and that's why we tell you not to use live vaccines like the herpes zoster vaccine, and we are certainly, even though you get flu shots and these types of things, in most cases they don't do much because your immune system isn't working very well.
Well, I guess, I'm curious about that, because if you look at some people they'll use a monoclonal antibody like daratumumab or ilotuzumab, and it'll work for them. And for other people, it'll work for a while and then it won't work for them, or the CAR T cells, sometimes it works, sometimes it doesn't. So I'm just wondering how you can segment, those people and who's going to work for.
Well, that's what the companies are hoping, that BCMA may at least make a stab at that. So the companies making a variety of monoclonals now are all over us to see if we can use the BCMA assay to predict responsiveness to immune oncology approaches. We don't know anything about it yet, but that's one of the hopes we have with this new test.
Okay, great. Well, we look forward to seeing it, and I think it'd be exceptionally interesting and wonderful to get this test in the clinic for patients.
We do too. And by the way,
Yeah.
it's very, very useful for CLL. We're working on lymphoma. It's certainly very, very useful for Waldenstrom's as well. We have a lot of data on Waldenstrom's and CLL now, and we're going to be publishing that soon.
Okay, perfect. Well, I have some other questions for you just because of what came out of ASH and the presentation that you did. So if you don't mind, we'll move on to a different topic.
Sure.
But your ASH presentation was on Kyprolis, which is a proteasome inhibitor, and you were studying how to dose Kyprolis. So what did you learn from your study about the dosing?
Well, it's very interesting. So we've been involved with proteasome inhibitors since the early days of Velcade. And what we had demonstrated in our early work with Kyprolis, that this was a drug that could overcome resistance of patients to Velcade. Velcade-resistant patients would respond. And the company had told us that the drug would have to be given 2 days in a row weekly, and that was based on a rat colon cancer model in which the drug worked better twice a week than once a week. And I told them, in the Berenson way, well, the rats lie, but the clinic doesn't. Let's do a trial. So then we embarked on a trial to give the drug once a week, and the drug was incredibly active, if anything, I think more active. And we have finished that trial. We're just getting that, I hope, published with a final review on the revised manuscript into blood. We presented that at ASH that in patients who received the drug 70 milligrams per meter squared over 30 minutes, rather than the conventional 27 for 2 days, twice weekly, the response rates look to be higher. Now, we even went further. So we're currently conducting a clinical trial, and I hope that we can get some interest in some of the audience and friends and them that may have myeloma to, believe it or not, take people who were failing twice a week dosing and simply take the drug instead of twice a week, once a week, and by God, it's working. I've never seen that in doing oncology in 40 years, that you could actually just change the schedule like that and get activity, and we are seeing it. So we are very hopeful that the future of Kyprolis will be a once-a-week administration, which we believe will be more active, and so far we're certainly not seeing more side effects with that more convenient dose or schedule.
And why it's very exciting. And so why do you think that is, that you can fail it at twice a week and then you can get it at once a week and then it's working for you?
Well, in the lab, with the higher dose, you get a higher concentration. It's called Cmax, maximum concentration of the drug into the blood, and that has more profound blocking on the proteasome and longer. So you get more sustained and deeper proteasome inhibition with once a week dosing rather than twice. You actually get better. And that may be the reason why. But God, it's so striking to see someone who's just completely failing on twice a week and then you go once a week, boom. I mean, that's pretty cool.
Yeah, that's amazing. Totally amazing. So in some of the studies, I know they're testing now doing Velcade versus Kyprolis, kind of head-to-head studies. And in the Endeavor study, it looked like they were comparing Velcade, Revlimid, and Dex with Kyprolis, Revlimid, and Dex, but it looked like the Kyprolis was a higher dose. So I'm just kind of wondering, is that an equal comparison? And can you take a drug, a similar proteasome inhibitor, and change up the dosing and do the same thing?
Great question. So it's interesting that Velcade did what we call MTD dosing and never backed off. So they did their 1.3 milligrams per meter squared, and then they never got off their high horse with a lot of their neuropathy. On the other hand, the carfilzomib Kyprolis folks, they did what we call an MED dosing, minimally effective dose, and they quit development at 2027. But clearly, this drug has a lot higher dosing it can go to. You know, we're doing 70 once a week. This trial did 56 twice a week. So I would tell you that you can push the drug higher. The problem with Velcade, you can't push it higher. So you could say it's an unfair comparison, but if you can't give more of the drug, in this case because of neurotoxicity, then you can't give more of the drug. So I don't think it is an unfair comparison. If the dose of the drug that you are administering in the experimental arm, in this place Kyprolis, is well tolerated, who cares that it's higher than the label dose? The label dose, frankly, was too low.
Well, that makes sense. And I know there are other trials coming out to compare, and I'm sure there will be others that will compare the other new proteasome inhibitors like ixazomib and others that don't have the same neuropathy issues.
Right.
So
I think I should chime in there though that the neuropathy with Velcade can be greatly reduced by using lower doses. We do not use 1.3, we use 1. And we give 4 doses a month, not weekly. Some people do it that way. We give it 1, 4, 8, and 11. And obviously the sub-Q. So I think if you give less drug, less doses per month, and sub-Q, you really greatly reduce the risk of neuropathy with this drug, Velcade. And you bring it into really Kyprolis range.
Interesting. So do you want to talk for a little bit about that? Because I know that's one of the things that you focus on the most is the dosing and administration of these different drugs. Maybe you can talk about first the existing drugs that we have or have had and how you do that, how you manipulate that. And then with the new drugs coming in, now what do you do?
Yeah, those are great questions. So let's talk about the tried and true. First, I mentioned Velcade. We give less drug, 30% less. We give 4 days a month rather than 4 days, 3 weeks. For example, with the chemo agents, with the alkylating agents, the Cytoxan, the melphalan, we give about a third to a quarter of the dose of cyclophosphamide and Cybor-D. We use a lot less drug. We only give it the first 4 days of the month at bedtime. We combine it with vitamin C because vitamin C actually makes the alkylating agents work better. Although you've heard that vitamin C, which is true, if you mix it with Velcade, it'll interrupt Velcade's activity. So we have them take the Velcade in the morning, the vitamin C 1 gram oral at dinner, and the alkylator at bedtime. And we never have, it's a very well-tolerated regimen. It's about 30% of the dose of cyclophosphamide and Cybor-D. Doxil, we've done a lot of work on Doxil. We've shown that low-dose metronomic Doxil, so the opposite of what we've done with Kyprolis, giving instead of 1 dose every 3 to 4 weeks like it's done conventionally, we give Doxil at low doses on Velcade days, and it is great, and you don't get mucositis or sores in your mouth. You don't get hand-foot, which means you get sores on the bottom of your feet and the palms of your hands. You don't get any of that with low frequent dose, because in our animal models we've shown that low dose frequently both works better and you can actually get a lot more drug in than you do conventionally. Moving on to the IMIDs, we don't like the doses that are FDA approved. We think they're toxic. We will use the Revlimid at 10. We'll adjust the dose down with renal failure. We don't start with 25. POM at 4 is not well tolerated in my view. We use 2 or 3. And a lot of the effects that are observed beyond the effects on counts, especially neutrophils with POM, are effects on memory, on mental function. Had several patients in this morning who volunteered to me their inability to keep their memory, to mentally function as well on the drugs at these higher doses, which have been required to be given by the clinical trials these people are on. So I'm a firm believer that less is more. We know these drugs work and dance well together. They synergize. So why not use less of them, not more of them? We're all about that in our clinic.
And your goal is to help patients control their disease with the best quality of life for as long as possible.
Yeah, I mean, I think the ultimate goal here is to do what my good friend Gio says. My wife's an actress, and she was in a lot of theater he did in L.A., and he used to say to her and the other actors on stage, it's not about more, it's about being more specific. And our whole thing here is to kill the myeloma and let you live your life. So we like to say it's not about complete response, it's about complete life. We do have data, though, like other sites, that complete response is associated with a longer survival. But in our clinic, it doesn't matter whether you achieve it in the first, second, third, fourth, fifth time you get a treatment. So we don't necessarily throw everything at you with the first regimen. We don't think that's the right approach. We do believe that is the right approach if you have high-risk features such as kidney failure where you don't want to stay on dialysis, so you really got to get a quick response, or you come in with plasma cells in the blood, so-called plasma cell leukemia, or the myeloma is already spewed out of the bone marrow and, say, into the liver or the lungs. That's really bad. Or if you don't have any blood counts and the bone marrow is totally myeloma, you got to go after it. But that's only about 15 to 20% of myeloma. So a lot of myeloma is a take your time kind of process. And I think we have to be very careful about over-treating. I can't tell you the number of patients I've seen in the last month who were beginning therapy and didn't even need treatment for myeloma.
Interesting. So with all the talk about MRD status, let's go back for a minute. Does the BCMA indication have anything at all to do with minimal residual disease? Do you test for that in your clinic, or what do you think of minimal residual disease?
Well, I think that, number one, BCMA will never be in and of itself a marker for that. It may be associated with it if you're in the normal range. But as I said, BCMA is present on normal plasma cells. So we don't have a marker that will only target the malignant population, which you have if you actually sequence the gene, which is what my work was at Caltech when I started my expedition in myeloma in the early 80s. If you sequence the immunoglobulin gene, the genetic material that gives rise to the antibody, you have a tumor-specific marker that you can measure minimal residual disease. The problem with MRD assays, they're only as good as the sensitivity and the specificity of the assay. And having worked with my twin brother on his first company, my twin brother, some of you know, is a biotechie who invented a device that would select healthy stem cells out of the bone marrow, and we did a lot of work on that. In order for the FDA to buy off it was something that worked, we had to show that in every single patient we could measure one tumor cell in 1 million normal cells. So we had, on every single patient, develop specific conditions that would allow us to do that. Nobody does that to measure MRD today. So you have slews of patients out there who are MRD negative and still have a monoclonal protein, which to me tells you how bad the assay is. So I think a lot of these assays are pretty suspect and not very good. It's tough, it's expensive, and I don't necessarily think that's going to tell you that much right now. I think ultimately it might, but I don't think right now. And the other thing is, a lot of people use bone marrow as the source of material to measure, and I already told you earlier on how heterogeneous the involvement in the marrow is. So I'm looking across the wall at the wallpaper, and it's all the same color. But I also have a lot of pictures on that wall, and those pictures, if you will, are like having variable amounts of myeloma. So you can put your needle in one part of the bone marrow, it's 10%, next door it's 0. So I think that's problematic. I really think that's a problem with the assay in a myeloma patient, to use bone marrow as your source to measure MRD.
And in the bone marrow, is it you're talking about quantities, you could get 10% or 50%, but also is there a difference in the type of myeloma cell, or is that pretty consistent no matter where you get the bone marrow biopsy drawn from?
It's a great question. I don't think we have a clue. I really don't think we know. Are there hot spots of myeloma in terms of what you talked about earlier, stem cell populations and other parts of the marrow that aren't so plentiful with them? We don't really know anything. We don't know the answer.
Interesting.
There was a little hint from one study that was the case. It was different in different parts of the bone marrow. There was one study.
Well, I know that other doctors have said that, on average, a patient might have up to 5, on average, different kinds of myeloma cells in their bone marrow. So I'm just wondering if that's floating around all the time, or if there are different spots with different genetic features.
I think it probably does vary. I think probably all the clones are probably present, but maybe in one spot it's 80, another spot is 20. But I think we're really not at a point to do a very good job of looking at that, because it's hard to get patients to give you 5 bone marrows, as you know. It's hard enough to give you 1.
Yeah, and give me conscious sedation, okay? So that's what I'm excited about. Well, I want to open it up for caller questions. So if you have a question for Dr. Berenson, please call 347-637-2631 and press 1 on your keypad. And we'll start with our first caller question, 9836757. Go ahead with your question.
Good afternoon, Dr. Berenson. Thanks for taking my call. This is Dana Holmes. I'm a smoldering myeloma patient, and I had a question about your BCMA assay. Is this actually detected in smoldering myeloma patients? And if it is, could it be used, or will it be used as a progression risk biomarker?
Yeah, in fact, that is a really great question. And we know, number one, it definitely is a marker for smolders. So normal's average 35, MGUSR's 54, smolders 85, and untreated myeloma is 520. So you guys are a little higher than MGUSR's. The big $64 million question is, if you're an MGUSR with, say, a 500, you're more likely to need therapy more quickly. We have data suggesting that, but we just don't have enough patients to answer it clearly yet.
Mm-hmm. Are you incorporating this assay while monitoring your particular smoldering patients?
Yeah, we are, but we're only using it as a research tool at this present moment. I think it will prove to be just as robust as in the actives, where if your level's higher, you're not going to live as long. So I think it will predict. And certainly the other telling thing, and it's really great data, in chronic lymphocytic leukemia, which is a lot like smoldering myeloma, the time to requiring treatment is profoundly different if your BCMA is above versus below the median.
Mm-hmm.
It's incredibly different. It's like 8 months versus 60 months. So if you had a high BCMA, you're really going to move quickly on. And by the way, with the MGUS patients, we don't have enough data yet to say the same thing, unfortunately. But I can tell you that anecdotally, the patients who've gone on to develop myeloma were ones that had the highest levels of BCMA with MGUS.
Mm-hmm. Mm-hmm. That's interesting. Do you have any trials for high-risk smoldering patients in your practice?
No, we currently do not, and I saw several this morning, and I certainly am very opposed to treating these patients with lots of toxic treatment when I follow them like a hawk. I don't think that you're benefiting them by intervening earlier, and I'm opposed to this relative risk being such that, hey, I'm going to go give this patient toxic therapy when I had even some this morning that are 10 and 12 years out and never been treated and probably never will need therapy.
Mm-hmm.
So I think it's a percentage game, but if I were a myeloma patient, I'd be willing to sit on the sideline and let all the other patients fight it out, because I think this thing is going to be cured in not too distant future. And why the hell do I want things like Pomalyst and Revlimid that are going to mess with my mind when I'm going to hopefully live to 92?
What about some of the antibody trials that are out there for the high-risk smoldering myeloma patients? Are they as toxic as the drugs that you just mentioned?
Well, ELO is certainly completely non-toxic, home run for that, but unfortunately ELO has no single-agent activity. DARA has a little...
I think the trials for smoldering incorporate Revlimid with that.
Yeah, well, there you go. Mm-hmm. There you go. And the data on... Darzalex, although 29% single-agent activity, a lot of infusion reactions,
Mm-hmm. Mm-hmm. Dr. Berenson, how do you follow? You mentioned that you follow your smoldering patients very, very closely, and that's your preference. How do you follow a smoldering myeloma patient? What tests do you include? Are there routine tests? Do you do annual imaging, things of that nature?
No, I certainly don't do anything like that, but I do 100% want to be doing tests in which the patients will be getting, if they're coming in for Zmet every month, a lot of them are, then we're going to get, every month we're going to be looking at the tests we do routinely and add BCMA into the test, although we only do that quarterly. But we certainly do follow the BCMA, the serum protein electrophoresis, quantitative immunoglobulins, serum-free light chain. If they don't have a lot of urine protein, we do a spot urine. If they do, we'll make them do a 24-hour urine, and they bring them in when they come in for their Zometa.
And what about Dr. Berenson? If you're putting a patient on Zometa, how do you establish the need for it? What testing are you using? Do you do DEXA scans at least, or any sort of regular conventional X-rays to determine the level of bone decline, if there is any?
Yeah, that is excellent question, and we routinely do bone density in anybody who shows no lytic lesions. We'll also do them in people who supposedly have lytic bone disease in the skull, because a lot of that's the figment of the imagination, the radiologist, and they're really being legs. And we don't. A lot of my colleagues treat anybody who has a hole in their head with a bunch of toxic treatment we don't do that. So if you have skull-only disease, we're not going to necessarily throw in all this garbage, the whole garbage dump into you. We're going to try to take it low and slow. We don't treat those people. So skull-only myeloma is usually not real. A lot of that is a figment of the imagination of the radiologist who sees on the form it says myeloma, rule out, and then they always find a lytic lesion. And a lot of times they're not real. I cannot tell you the number of patients I have who come in to see me and have been on toxic therapy and even have MGUS and are told they have myeloma because of imagined skull lytic lesion or other lytic lesions. I have our radiologists read them, are really good, that I deal with all the time. I've been reading myeloma x-rays for 25 years, and they're like, there's nothing here. And it's just really frustrating.
Yeah, it's really unsettling to hear that as a patient. It really is.
Yeah.
So you don't use routine imaging, but you do use imaging initially to evaluate, I would presume.
Yeah, and then like I just saw one of my smolders today, and every few years, to keep me honest, I'll get a bone density on them to see if they move to required. So if they have a bone density that's worse than -1, then I'm going to go ahead and shove into these people Zimet every month.
Do you you obviously do bone marrow biopsies on smoldering patients?
I'm sorry, say that again?
Bone marrow biopsies?
Well, we only do it initially. I don't routinely do it every.
Right.
Yeah, I just do it initially, but I don't do it unless there's a change in clinical status.
Okay, and
I'm
what
not going
about
to do it.
What imaging do you do though up front? The skeletal surveys, or do you use
I don't
MRIs?
do PETs and MRIs and all this
No?
stuff. I mean, I'll do if you have a solitary plasma cell tumor, I want to know if it's systemic, so I'm going to do a PET CT on those people. If you have a patient who has symptomatic disease in the back, and most people do have back pain from being just older, I'll do an MRI to look and see whether it's degenerative disc disease, spinal stenosis, or myeloma.
It's a very personalized approach.
Yeah, we don't assembly line myeloma here.
Yeah, that's nice to hear. Well, Dr. Berenson, thank you so very much for your time. Very much appreciated. Thanks, Jenny.
Okay, thanks so much, Dana. Okay, our next caller is at 371-3695. Go ahead with your question.
Hey, Dr. Berenson. I'm just trying to nail down this alternative Velcade administration. You use 1 per meter squared, 1, 4, 7, and 11 Q month?
Yeah, 1, 4, 8, and 11 every 4 weeks.
Got it.
And then when you're and then when you're done with the treatment, usually 6 to 8 cycles of that, we go to every other week maintenance, and we give 1.3.
Okay. So when do you determine that that's the time to make the switch?
Well, once you plateau your response, but we usually go between 6 and 8. So, your IgG goes from 4 to 2 to 1 to 500 and then sits at 500. We usually don't give less than 6 cycles, usually not more than 8. We like Dick Van Patten, so we say 8 is enough.
Okay, so then so you would go ahead and plateau and then go to 1.3 per square meter per week?
We would. I'm sorry, that went in and out for a second. The phone. Would you say that again?
You would wait for the plateau and 1, 4, 8, and 11.
No. The initial therapy is 1, 4, 8, and 11 at 1, whether it's with Dex or Doxil or whatever. That's what we do. Or Revlimid, and then once you plateaued out, we usually do that treatment plus 1 or 2 more when you plateau. And then we go to every other week at 1.3.
Every other week, okay. And have you published any of this head-to-head?
Oh yeah, we published 1 million articles on alternative Velcade dosing.
I know you have, but in this particular.
There's no good data on maintenance at all. Zippity doodah. I mean, there's just nothing on that maintenance does anything with Velcade.
Got it. Thank you so much. Bye.
Okay, thanks so much for your question. The next caller is 847-5748. Go ahead with your question.
Berenson, what is your view on transplant for patients that have achieved VGPR through chemo, 20 cycles of CARP on Dex? And what is also your view on the timing of the transplant and its effect on overall survival?
Yeah, well, I don't transplant anybody anymore. I'm a guy who trained under the guy who won the Nobel Prize for developing him in Seattle at the Hutch. But I don't think that's a modality you need to use anymore. We have so many options today. I would say that maybe 1 out of 30 patients benefit. I'd say 1 out of 30 it actually makes the disease worse. I can't tell who the 3% who are the winners, so I'm not going to do it to not benefit 97%. So my timing of transplant is don't do it. Occasionally, end stage, I may have a patient who goes for transplant, but I think it's a fairly toxic, ineffective program, although somebody who's listening to this I know has done well by it, and she may be one of those few percent. And I do have some of those people who were actually part of my brother's CD34 selection trial in the late 90s who are still doing well. But I have patients who were on Velcade trials back from the early 2000s too, with beautiful complete remission. So, I'm about, it's the long game here, not the short game. But I think there may be a small percentage of people benefit, but we don't do it. We don't recommend it.
And is there any a priori way to determine, either through genomic studies or some type of correlation?
If there is, nobody knows it. If there is, nobody knows the answer to that.
Understood. And even in a relapse setting, you
yeah, I don't do it ever.
Understood. Thank you.
Yep.
Okay. Thank you so much. And I think it would be ideal to just have patient-reported outcomes with a lot of data, so we can see who does what with the best, and then we know what to do.
Well, you know what, Jenny? Music to our ears, because we're just about to publish our data on our own 300 patients in 2 separate articles, one on specific drugs. It's beautiful data on everything that's ever happened with these folks, and also looking at our survival curves, which are much to the delight of my patients. So I think groups should have to do that, and we are doing that. And the really interesting, provocative thing I'll tell you before closing is that the response rates to these drugs keep on trucking. They don't just one and you're done. So don't believe for a minute because you've failed Velcade, say with Cytoxan, it won't work with Revlimid, or that because you failed Velcade, it won't necessarily work with Kyprolis, the same drug. There are so many combinations now, it is amazing.
Well, that's great to hear. So, well, Dr. Berenson, we are so glad you joined us today and are learning more about your test. We look forward to seeing that come out. We're just so grateful for your incredible research and your efforts on behalf of all myeloma patients. We're just so thankful for all you're doing.
Well, thank you, Jenny, and I hope you enjoy the snow up there. Hopefully I'll get up there in March to ski with you or you and Anna.
Okay, sounds great. Okay,
all
thank
right.
you so much.
All right, thanks a lot. Have a great week.
Thank you for joining us on Myeloma Crowd Radio. We encourage you to get fit with us and join the Muscles for Myeloma Fitness Challenge.
It is Ryan here, and I have a question for you. What do you do when you win? Like, are you a fist pumper, a woohooer, a hand clapper, a high fiver? I kind of like the high five, but if you want to hone in on those winning moves, check out Chumba Casino. At ChumbaCasino.com, choose from hundreds of social casino-style games for your chance to redeem serious cash prizes. There are new game releases weekly, plus free daily bonuses, so don't wait. Start having the most fun ever at ChumbaCasino.com.
No purchase necessary. VGW Group. Void where prohibited by law. See terms and conditions. 18+.